Hello, everybody. So nice to some of us be in person again after this break. We have plenty of people online too. And for those who are online and for those who cannot join us, we're also recording and we're going to make everything available on our YouTube channel so you can actually watch some of this stuff if you need to leave early or if you can only be here for a little bit. I was also told that Subway is down. So there are people who probably will be joining us late. But what I thought I would do today is spend the first half hour of the meeting to talk about the last 10 years, because we've made it to a little bit over 10 years now. And as you heard from our OBI Representative Jordan, we have been renewed for potentially another 5. So 3 plus 2, we're planning for 5, because it depends on how the government issues the contracts. And so it's probably a good moment to take stock, right? Stop and see where we started and what we have achieved in the last 10 years as we're having conversations about where to go for the next 5 years. So let me see if I can figure this out. And I'm going to put glasses on because I'm getting old. Okay. So I have some disclosures, you guys know, but I always do that. Not because there's something wrong with us having lots of partnerships and lots of funding and partnerships with both federal and provincial and industry, but because it's important for us to understand who funds our work, because there are biases that come sometimes from implicitly without us thinking about it in terms of who is invested in the space, who gives us money, and who we actually report to. So I always put it there, but if you have questions about that, we can talk about it later during the day. So this is where we started. So the challenge was more than 300,000 kids with neurodevelopmental differences in Ontario. Sometimes we have some good interventions, sometimes we don't, but most of the time, we have not been able to change the ultimate developmental trajectory. So we haven't been able to get the kids on a different course of development, but is more likely to meet their versions of a good outcome, their versions of what a good life is in adulthood. So we took a step back to think about why that may be. And there are many, many, many reasons, and we can have that discussion later, but of relevance to our network, one problem was that there is a lot of variation, a lot of heterogeneity, variation within each diagnostic label. So 2 kids with autism can look very, very different. 2 kids with ADHD can look very, very different. 2 kids with OCD can look very, very different. Kids with intellectual disability look very different from each other. Even when we do rare genetic syndrome, so think about Rett syndrome, Fragile X, some of you are in the room, you see 2 kids with the same genetic condition and they look very, very different. And part of the problem here is that we have assumed that just because they have the same label, even though they look different, they must share the same biology that underlines their differences, and therefore we have grouped them together in clinical trials and tried with a single intervention to actually make a big difference. But the truth is they probably have very different biologies, and that's why they look different, right? And understanding what these differences are across all the different diagnoses, no matter what we call it, may be very critical to us being able to match a right intervention to the right kid, what we call in precision health or personalized medicine, right? So the labels are useful for all kinds of reasons, but ultimately, to be able to achieve precision health, to be able to achieve personalized medicine, we need to know that child in front of me right now, not some average child in a study. What is their underlying difference in their brain, in their body, in their social determinants of health that predicts how close they are to their version of a good life, of what they want as a good outcome, and how do I go after that child's particular characteristics to be able to have a big impact, right? So to do that, we understood that we need to understand the differences across diagnoses better. We also understood, and so that we can create groups of patients who have very similar biological differences. So when we get interventions targeted at groups of kids or adults or youth, we know that that group of patients shares a lot of their same biology. There's some other problems in our ability to actually change outcomes. One has to do with actually not having it very obvious what the target of intervention should be. So within a condition, we get many different biologies. So if you think about biological targets for intervention, we need to think about the different biological targets we have. Some of them are targetable, if you like. We can go after them. Some of them we can't. But also, when we ask youth and families what they prioritize as an intervention target, we get many, many different responses. So when we design studies, it's very hard for us sometimes to figure out what the actual intervention target should be, right? This is where we started. So I'll show you what gains we've made. We also have some regulatory things we can talk about later if you're interested that are challenges. So I start usually from this slide. I have kind of made the case. The slide is based on putting autism in the center and other conditions on the side, but you can do the exact same thing for every condition that's in the room. You can put ADHD in the center, everything else on the side. You can put OCD in the center, everything else on the side. The idea for this one is this. When a 2-year-old presents for an autism diagnosis to a clinician, if they get the autism diagnosis, chances are they just have autism. But for those of us, I'm a clinician and I'm a scientist, right? So I see kids across as they grow and they become teenagers and they transition to adulthood. As these kids grow, it's rare to find a 16-year-old that just has autism. They have a little bit of ADHD, they may have a little bit of anxiety, they may have a little bit of depression, they may have sensory and motor differences, they may have epilepsy, they may have gastrointestinal differences, they may have immune differences, they may have sensory issues, they have a variety of language abilities, they have a variety of cognitive abilities. So as they grow, they keep accumulating these labels that we think of as distinct. Same thing for ADHD, right? So when a kid gets an ADHD diagnosis, first, chances are it's because that's the most prevalent symptom. They're hyper, they're inattentive, they're impulsive. But as they grow, people realize that they actually start collecting diagnoses of other mental health conditions. And about 20% of the time, as they grow, we're like, are you sure it's just ADHD? Because they have quite a bit of a social deficit, and is it time to actually start thinking about whether they have social impairment? And obsessive compulsive disorder has a very similar profile also, and intellectual disability even more, right? Because typically we make the diagnosis of intellectual disability just because somebody's thinking skills are sitting below a particular cutoff, but it means nothing in terms of what is the underlying biology. So depending on their underlying biology, sometimes they have autism, sometimes they have ADHD, sometimes they have OCD, sometimes we find them, they get mood disorders, they get anxiety, and so on. So if that's the case, then what are the different categories that are meaningful to target with interventions to make a big difference in the lives of kids, right? I put on the side the other three to remind us that also there are some other biological processes that make it much more likely for a child to have a neurodevelopmental condition. So for example, if a child is born premature, the likelihood that they're going to develop a neurodevelopmental condition is between 5, 10, 15, depending on the condition, times higher than that of the general population. If a child is born with congenital heart disease, the chance of having a developmental condition is sitting in the 30 to 40%, right? Heart disease, chances of having a neurodevelopmental condition, the vast majority, quite a bit of a third of the kids will actually have a diagnostic label, and another third will have impairment in certain areas, although they don't meet criteria for a diagnostic label. And of course, you're all familiar now with genetics. So certain set of types of genetic variation would make it much more likely that somebody has a neurodevelopmental difference that actually is associated with impairment. So how do we think then about these categories if kind of things that have nothing to do with a neurodevelopmental condition per se, that are biological and early on in fetal life, predict the development of those neurodevelopmental conditions. So I'm going to skip the picture because I have, come on, I've presented that family before to get to what it is we built and what it is we did. So what we did is we said, maybe we are wrong. And sometimes it's important to say that, right? Sometimes the scientists and the docs get it wrong, right? So maybe the way we classify kids into these neat little categories that are called autism, ADHD, intellectual disability, OCD, Rett syndrome, whatever have you, is not the right way to do it. But then, if that's not the right way to do it, what's the right way to do it? Well, we're scientists, we can think about ways to collect data and use advanced mathematics to help us figure out what the right categories may be. Right? So what we did is we created this network that goes from Queens to SickKids and Holland Bloorview in Toronto, to McMaster, to London, and I saw from the responses that we have participants in the meeting from all the sites. And then we said, we're going to confirm your diagnosis, so everybody knows that if you came in with a particular diagnosis, it is what people think as a diagnosis. So you all got diagnostic testing for the condition you presented in. But we are also going to become a little bit agnostic. So agnostic means we're going to accept that we may not know. And we're going to collect other types of data, not in a way that's specific to your diagnosis. So we're going to collect information about your genes, and we're going to collect information about your brain structure and your brain function, and we're going to collect information about some markers in your blood that may be relevant to immune function or endocrine function or hormones or things like that. And we're going to actually measure your behavior in terms of how the parents actually see it in real life, and in some circumstances, we measure it directly for certain things, and we're going to measure your thinking skills and the way you kind of problem solve, and then we're going to use all that information to try and use computational methods, advanced mathematics, to try to understand if there are groups of kids who are much more alike each other in terms of their brain structure, their brain function, their genomic underpinnings, their behavior, irrespectively of those labels, to help us understand, do these diagnoses actually matter, or for the purposes of understanding how to do personalized health, right? They may matter for other things, but for the purposes of doing personalized health, or should we rethink it? And then we built a clinical trials network that you heard from Jordan, which was the first of its kind in Canada, and we embedded this clinical trials network into our bigger, what we call a biomarker core, so that when we do intervention studies, first of all, we pick targets that come out of biology, so sometimes genomics and sometimes other systems neuroscience. Secondly, we use the treatment response to try to understand what these groupings of kids are. Again, we're in the process of this. This is not a short-term game, but I will show you where we are. So first of all, we can only do this because you're here. There are 4,000 families, more than 4,000 families now in the consortium. So thank you, and I mean that, thank you. There is no way that this kind of work can be done without the participation of families across the conditions, across the cities, across all kinds of other diversities that we have, and I can talk about that later, to actually help us understand the issue. There are more than 30 investigators. We have a very active patient advisory and youth advisory. And for those of you who are not part of our patient and youth advisory, this is an open invitation. This is an open group. You can join us anytime you like, and we're actually revisiting a little bit how we are, their different roles, and how we actually make it possible for families to participate, because we know it's not easy for everybody to keep volunteering their time. So we're changing the way we are doing our patient advisory so that people get compensated for their time and they get to choose the role they have that's appropriate for their family. So please join. How am I doing? Somebody needs to tell me how much time I have. Technically 10. No, I'll go for 10. Because, no, so I'll show you what we've done, And then I have all the next steps, but maybe I'll keep the next steps for the end of the day, or I'll show you just a couple of things from the next steps quickly, because I know some of you are going to leave after lunch, just so you have an idea. So I'm not going to talk about genomics because we have a whole genomics talk. The only thing I'm going to say about genomics is you should know that the samples that you have contributed for genomics have become part of consortia that are first in the world in terms of understanding the genomic architecture of the different neurodevelopmental conditions. And there have been lots of lessons learned that you're going to hear later from our genomics, from Dr. Steve Scherer, who's hosting us at SickKids today and leads our genomics platform. The only thing I'm going to leave you with for this slide is lots of genes have been discovered, and then we care about genes because we care about processes that we can then think about targeting to improve quality of life for kids with neurodevelopmental conditions. We have gotten better at grouping genes together to understand these metabolic pathways that we can go after for potential interventions, and I'll show you an example of that we have already done. But we also do not find genes that are particularly specific to autism or ADHD. Like those genes seem to confer risk for any neurodevelopmental condition. And in fact, I put an example here that is not POND from Dr. Scherer's work. This is part of a Baby SIBS consortium where families who had an autistic kid first participated, enrolled their second kid when they were born, before they had any neurodevelopmental concerns. And they looked to what degree the original genetic difference predicts autism in the next child. So that the ability to predict autism in the next child is almost, flip of a coin. But the ability to predict any neurodevelopmental condition is pretty high. Again, we are learning that a lot of these original biological differences make it more likely that somebody develops different, but the exact kind of difference is very hard to predict from the beginning. But I will leave the geneticist talk to you about genetics, and then I'm going to give you a little bit of an update on what we did with imaging. So we started at just comparing the groups. So we started comparing, and don't worry about I'll show you what you need to see. We started comparing the groups in terms of their structure and function to see whether autism and ADHD and OCD are particularly different. There's many papers, and I put papers for you there, not because I need you to actually read the papers, but I want you to know there has been huge academic record that came out of this network over the last 10 years that is changing the way the field is thinking about. So I'm just putting them there for you to just have a sense for it. But I put those two papers as an example to say that whether we look at structure or whether we look at function of the brain, we were finding it very hard with large samples to find differences between groups, although there was a lot of variability. What does that mean? It means that on average, if you pretend that you have an average child, which we know doesn't exist, but that's what we do in research often, right? We average across. An average autistic kid, an average ADHD kid, and an average OCD kid. Between autism and ADHD, when we looked at brain structure and function, we found very little difference. And when we looked between autism, ADHD, and OCD, the front of the brain had very little difference. It's just that it seems like it spares the back of the brain a bit for OCD. But when you look at these graphs, I want you to look at these black lines. You see those black lines that go through the bars? We call those error bars, and it is a sense of how much heterogeneity, how much variability there was within each group. So although the average child looks similar across the groups, there were many different kinds of children in each group. So it is not that all of it is the same. It is clearly not all of it the same, right? Lots of kids with many different brains, structure, function, and so on. But the diagnostic label was not helping us tell who's different from whom. Yes?
What do the colors mean?
So this one is, purple is, let me just look. Again, I'm blind, I'm getting old. Autism is purple, yellow is ADHD and green is OCD, and the red bars are typically developing kids. This is across different networks, resting state, functional networks in the brain. So you have here, for those of you who follow this, oh, you cannot see my, oh, all this time I've been pointing, you have not been seeing my pointer, right? I'm sorry, do we have a pointer? You don't see a pointer. Okay, so I'm gonna talk through it, okay? So on the right, you see resting state connectivity, so how your brain is connected when you don't do a particular task. The green, the non-red, the non-orange color is the different groups, so OCD at the bottom, ADHD in the middle, and ASD at the top. The red group is the typically developing volunteers who come through our network so we can figure out what's different. And at the bottom, you have different networks. So, for example, SC stands for subcortical, so the kinds of messages that goes from the basic archaic structures of our brain to the cortex, where we do more our thinking. DMN is our resting state network, so the network that is most active when we are resting and sitting with our thoughts. Frontal parietal networks that are very important for a lot of the thinking activities that we do, for planning, for attention, for organizing. Limbic is the system that's very important for our emotion regulation. And then we have two attention networks there, the DAN and the VAN, dorsal and ventral, but they're attention networks. Sensory motor, the network, the SMOT sensory motor, and it's the network that controls how we take sensory input and how we execute motor activity, and then our visual network for these. So basically what you see there is that the pattern of differences between typically developing kids and whatever the condition is very similar, right? And in the first graph, we have the same kind of scenario, but for structure this time, and we're looking at surface area and cortical thickness, and the patterns of differences between typically developing kids and kids who have these conditions is very similar, but lots of variability, lots of different brains. So then we couldn't figure out, so when we got to that point, we had to figure out what this means, because there were two possible scenarios. One scenario was that in fact there are different biological conditions under all of this variability. They just spread out across the different diagnoses, and that's why we cannot get the differences in the average child. The other possibility, which is as good as any to make a hypothesis, is that this measurement is very, very noisy, that there is a lot of individual differences, no matter what, whether you have a condition or not have a condition, and seeing variability doesn't mean very much for biology. It's just part of the individual differences and the noise of our measurement. So we were trying to figure out, is it biology? Is there structure to this that we need to understand? Or is this noisy? And this is when we got our animal model colleagues to help us out. And some of you have seen this many times, but I'll highlight just a couple of points since I'm doing the tour of the last 10 years. So we took mice and introduced genetic variation that we see in humans who have neurodevelopmental conditions, and then we compared again, and then we imaged them the same way that we image humans. And we compared how the mice who had the genetic variation that is predicted to cause a neurodevelopmental condition in the human was different from a mouse that did not have it, right? To see if we get this kind of variability. So since I don't have a pointer, I'm gonna just ask you to use at the graph that just has the blue columns. And what you have on that graph is on your x-axis or at the bottom, you have all kinds of different mice that have different genetic differences. And on your y-axis, you have how different the mouse that has the genetic difference is from a mouse that does not have a genetic difference. So if there is no difference between the genetically modified mouse and what we call wild-type mouse, you should be at the zero line. You see the zero line in the middle? And then if you have much bigger brain, this is about brain growth. So if you have a much bigger brain, you're sitting on the right side of the graph. So you have some mice that have really big brains when they get genetically modified. And then on the left side, you have the opposite direction, mice that have smaller brains when you're genetically modified. The point here is that all of these mice, big brains, small brains, and everything in between, have genetic variation that's predicted to cause a neurodevelopmental condition, right? But they are quite variable, right? They are quite different from each other. And so if I were to take an average of this in the middle and put my little black line, my error bar that says how variable this measurement is, I would probably show you what it would come up as I show you on the human data, where we find a small difference between the typically developing kids and the neurodivergent kids, no matter what the diagnosis is, but a lot of variability. However, we know it's not noise because we know our x-axis. We know what predicts whether you're going to have a big brain, a small brain, or everything in between. It's a genetic variation, but sitting at the bottom. Right? Are you with me? Okay, so that means that we learned that although the average kid in between conditions seems similar, there are a lot of different conditions under their biological conditions, but are predicted in this case by our genetic variation that predict a different brain, no matter what we call it, right? It's not noise, it's just different biological conditions. With me? I see nods. I take the nods as a yes. So then this was in mice. If that is true, then when we go back to our human, let's say when we go back to that study that has the bars, we should be able to use advanced mathematics to figure out kids who have brains that are more similar to each other, that we can predict have a more similar biology, and it's not just noise, right? So we went back, and now you see just examples here. Again, Dr. Ajay Kuski sitting in the back. Woonjung now is sitting, she was in Toronto, now she's in London. So people have grown during this network, which is the other thing we don't talk about, but lots of people trained, graduated, and our faculty and have labs in other places. And then we clustered the actual human data now of the brains to see whether we get groups of people who have more similar brains, forget the diagnosis, and then looked to see if the diagnosis actually works. So I'm just going to do one of the two examples. I'm going to do the top one, where as a cluster, I think cortical thickness and volume, so the structure of the brain. And you'll see in that particular case, you got 10 clusters. Again, the number of clusters doesn't matter for our purposes right now. We can discuss this in the future directions later in the day. But what I want you to see is if autism is purple, ADHD is light blue, OCD is green, and yellow is typically developing kids. We only have a tiny little cluster at the end, but it's autism specific. But we have it. So there are some kids who have autism who are clearly distinct from everybody else. If you look at the purple bars, they're spread across 10 clusters. So kids with autism can be different in this particular scenario 10 different ways, and they group with other conditions. So you'll see on the right side what we call neurodevelopmental disorders group. These are clusters of kids who have different neurodevelopmental conditions. So they could have autism or ADHD in clusters 8 and 9, or in the clusters 7 and 6. They can have either autism or ADHD or OCD, but they are more similar to each other than they are with other kids who have the same label, right? So a kid who is in cluster 10 is more different from an autistic kid in cluster 7 than an autistic kid and an OCD kid in cluster 7. Does that make sense? Right? So our label is not helping us tell which kids have similar brains, because these clusters of the similar brains map to different diagnostic labels. Okay, and then I'm told I need to finish. So we spent some time, in this case, we didn't look at structure, we looked at functional connectivity. So how does the brain, different parts of the brain connect to each other and communicate with each other? And in our clustering exercise, we looked to see, first of all, whether any of the groupings that we get map to diagnosis. Secondly, what are the characteristics of those groupings? And even more importantly, because sometimes we hear ourselves talk and we're in echo chambers and we think that we see something in our data and we think this describes the rest of the world, and that's not true. We took a completely independent network in New York, the Healthy Brains Network, nothing to do with us, except it includes kids with autism, ADHD, and typically developing kids, and replicated. We did the exact same procedure in the completely independent cohort to see if the things that we are learning from POND actually generalize to kids beyond our cohort that may have some characteristics that may predict things that may be only special to us. So I'm going to say 3 things about this. First, I can tell you there is no diagnostic specificity. When we look at brain, no cluster had any diagnostic specificity, meaning every cluster included kids from multiple diagnoses. Secondly, the most important thing to the brain seemed to be cognition, so thinking skills. So the kids who have higher thinking skills and the kids who have more struggle with their thinking skills clearly have different brains. The next thing that's important to the brain is ADHD symptoms. So hyperactivity and inattention was the one that replicated between actually the 2 cohorts as a feature, as a symptom that has a big brain signature, right? And in our cohort, also some of the OCD symptoms, although not on the New York cohort. Then, in the next split, the most important thing was socioeconomic status. So we're going to act on this in the renewal because it says that the access you have to privilege has a big signature on your brain, structure and function. And after that, we got sociability. So for all the social stuff we talk about, it's actually the detail on the brain. Right? It comes after the signature of socioeconomic status. So things to act for the renewal. I'm going to say a couple of more things here because I know Alana wants me to stop, but it's important for the rest of the discussion. The other thing we did all together, if you remember, some of you, is we did a large exercise with very robust research methods across Ontario and across neurodevelopmental conditions to develop research priorities. Remember we said we don't know what to treat. We do not know what targets you want us to go after. So we had a large partnership with the Ontario Brain Institute, and we did a large exercise over about 2 years to develop our top 10 research priorities. And I will summarize to say what you have prioritized are a lot of precision health things. So you want to know which intervention for whom, when, how much, when to start, and when to finish. You prioritized 5, I think, or 4 symptom domains. So behavior, emotional regulation, anxiety, irritability, aggression, and self-injury, and social skills. And you also prioritized interventions for families. So navigation and maintaining the wellness of a family. So we've heard those, and you'll see that in the next steps and in the renewal, we'll act on those things. So the last thing I should mention, because it's critical of what we do, is the clinical trials work. So when we started looking at the biology as it comes out from genetics and the genetic differences that pop up for these neurodevelopmental conditions, we group them into potential pathways that can be druggable or targetable with technology or other types of interventions. Again, we're not looking to nobody's interested here in normalizing anything. We're looking to improve the molecular pathways that cause impairment and dysfunction, right? And so then we started mapping medications to the biological pathways and not the way we do it now in clinic, which we're going to talk about in the psychopharmacology breakout later, which is we see a symptom, we kind of guess how it's similar between conditions, and then we borrow drugs from other conditions and try to treat them. So the example that I'm going to give here, if I can move this, there we go. Because I'm out of time, I'm going to put them all up, is we did a series of studies that took a target from the biology, from what we were learning from genomics, from what we were learning from systems neuroscience, and used drugs that exist on the market or don't exist on the market. In this case, Riluzole exists on the market for Lou Gehrig's disease. Tidaglosib doesn't exist on the market, that have a target, a biological target that we think is of interest to our kids, although it has nothing to do with what they are used for in the market, right? It's nothing to do with behavior or cognition, the actual use that they have. And we look to see whether we do better in our clinical trials than when we actually take a behavioral target. So in this case, if you look in the Riluzole study, the blue line are the kids who took the sugar pill, the red line are the kids who took the blue line is the kids who took the drug, the red line took the sugar pill. So in the first one, this was in autism, the social withdrawal was the primary thing that people thought about. We showed a trend for decreasing social withdrawal in our drug that looks better than placebo but doesn't beat it statistically because we have a lot of variability. On the other hand, when we take a symptom that you guys prioritized in your priority exercise, which is externalizing behaviors, irritability, hyperactivity, we actually statistically separate, meaning we have statistical confidence that the drug works better than sugar pill for improving symptoms of irritability and hyperactivity. So we are learning from you that what you think defines your condition and is important to treat is closer to the biology than how we have defined the condition, right? So when we take a biological target, the thing that is most likely to change is the thing, but is the thing that you prioritize, not the thing that we define the condition by, which is kind of interesting, a very interesting learning. And then because we didn't have enough kids in our consortium, we partnered with the European consortium in the last few years and ran a study together with the Europeans. And I will show you now the results from this study, which is arbaclofen, and this you haven't seen before, so it's new. So arbaclofen was the last study we ran. It's a drug that goes after how noisy your brain is, because we learned from genetics that a lot of the genetic variation is in the synaptic space where the message is transferred from one neuron to another. So if we have synaptic deficit, we either get a lot of noise that drowns our signal in our brain, or the opposite, we kill our signal. So between signal and noise, we cannot tell the difference. So this drug is supposed to decrease the noise in the brain. And so we look to see whether it moves social skills, and we look to also see whether it moves social and repetitive behaviors in autism. And you'll see we got a trend for improvement in social skills, but we got very significant improvement in sociability and repetitive behaviors, and I can tell you the Europeans found the same. So now we're pulling the data to have a pre-specified analysis, and we're going to partner with Dr. Scherer and the equivalent of Dr. Scherer in Europe to look whether some of the genomic architecture actually predicts treatment response in this consortium. I'm going to skip this because I'm out of time. So I'm going to finish with the other quick hits from the last 10 years. I'm going to put them all in one slide. And then we're going to talk about future directions at the end of the day. So for quick hits, our anxiety meter, some of you participated, was the device that we developed to pick up emotional dysregulation and arousal dysregulation and how it links to anxiety. So we have a patent, we have a device, and you're going to hear from Azadeh later during the mental health session about where we are with the development of the anxiety meter so that we can get it in the hands of kids who need it. It's already in the hands of adults who need it. That's like a heads up. And we're working to get it in the hands of kids who need it. The second thing is we used our clinical trial infrastructure and expertise that we built to run a large virtual clinical trial, not of medication, but of anxiety intervention, group-based anxiety intervention virtually during the pandemic. I put up the map to show you that we got all over the province. Not only we ran it all over the province, but we trained therapists all over the province to run it. So when we walked away from the trial, we left the expertise in the communities where we ran the trial, which was positive, has been reported to the Ministry of Health, and we're working on a model to get it publicly funded through the current publicly funded system. We collaborated with our partners around the world during COVID to understand how COVID was influencing the mental health of kids. Some people here had leadership in this particular consortium, like Jen. We linked your data to the administrative record that the province keeps to start understanding how real-life outcomes, like how often you end up in the emergency room, link to the biomarkers that we actually collect, such as the genomic architecture. And you're going to hear from Danielle from this later, so that's why I'm not going to present it. So she's going to show you how we are starting to understand how genetics may actually predict your real life things, not just the things that we measure in a research study. We shared our study in the public domain, so other people can actually come and use it to actually ask the questions we cannot ask. I mean, there's that many hands around. And frankly, we all have a way of thinking, and we want people who think differently to be able to come and access the data and actually challenge us on what it is that we are learning. We partner with our youth to develop a project of digital mental health stories to tell the story of their mental health in the system. And I can tell you right now, and I'll give you more detail later, but we have partnered with UFT's Interprofessional Education Center to actually develop curriculum based on those stories for across health professions and students to train on how to deal neurodevelopment and mental health. And we have partnered with industry and other consortia, and I just put the Digital Supercluster project as an example of those. We have many, where we pool our data, our brains, some of you are participating directly to actually ask much bigger questions than the ones that we can actually ask ourselves. So what's next is my next slide, and I'm out of time. So what I'm going to do is, when we talk future directions at the end of the day, I will show you everything we're planning to do in the next 5 years, what it is that we still have to do to decide with you, to partner, to actually make decisions on, and what the potential of the next 5 years is. But I just wanted you to have a sense of what the last 10 years look like, where we started and where we are. There will be plenty of time for questions later, but if there is one or two, maybe I can take now, and then I'll pass it on to genetics.